everyone, welcome back to the channel. I'm the GCSE science teacher, and in today's video we're going to be discussing a required practical for GCSE biology. It's all to do with food tests. Comment down below if you have been taught this recently or it's coming up and you want to revise it. Perhaps you're doing a mock exam. Let me know down in the comments. It's always good to hear from you. And if you do find the video helpful, feel free to like it, share it with someone you think would also benefit from learning more. And you can find more revision resources from me for GCSE biology, chemistry and physics on my Instagram and TikTok at the GCSE science teacher as well. Thank you so much to everyone who's already subscribed. It's great to have you as part of our community here on YouTube. So let's get started. So, the aim of this practical is to use qualitative reagents to test for a range of carbohydrates, lipids, and proteins. And one thing to be aware of is this is a qualitative method. So, we're looking for a color change here, and that will determine whether there is a type of food molecule in a certain food substance. Unlike other practicals where you're actually quantifying your results, you're using data and graphical analysis, you're actually using a subjective analysis. You're determining whether there is something present, yes or no, based on a color change, something that you observe, which can actually be helpful, but it can be subjective, like I say. So some people may think, oh, that's green, and someone might be like, I think it's more blue, and that can change the dynamic of the answer. And that's one of the pitfalls of this practical. So it's something to be aware of. Something also to be aware of whenever you're talking about practicals, you need to know about the variables. So let's quickly run through them. A control variable is something that you keep the same, and the reason we do this is to compare the results later on in the experiment. So in this particular example, we would keep the same, say, the type of food we're using. Let's say we're testing a loaf of bread, and we're going to use the same loaf, the same slice of bread for every single reagent. And that means that the results are going to be more accurate and therefore reliable as well. We've also got the independent variable, which is something that you're going to change or alter. And that is important because we want to change what we're looking for sometimes. So, for example, we might want to change in this example. We're changing the reagent we're using. So we are looking for carbohydrates, we're looking for lipids, we're looking for proteins, and therefore we're going to need slightly different chemicals or reagents. Okay. And then we also have the dependent variable, and this is what we measure. So in this case, we are looking for a color change. That's what we're observing. But in other practicals, we might be measuring the volume of gas released, or we might be measuring the temperature change. It just depends on what practical you're doing. So be aware of the three different variables and try and keep condensed notes on what each of the variables are for each of the required practicals. So, the equipment or the apparatus that you need for this practical, there is quite a lot. And if you have done this in class before, you may have been asked by your teacher to work in groups and separate what each group is doing and then collaborate with the data at the end. That's something I've done in the past because it's a lot of steps and a lot of different bits of equipment that you need. And obviously, depending on how much time you have for this practical, will depend on how far you get. So, some of the things you need in terms of glassware, you're going to need a conical flask to help you collect the filtrate. And I'll explain that in a moment. That very much links with the filter paper and the funnel. Remember, a funnel needs to be placed inside a conical flask and not a beaker, because obviously the neck of the conical flask will hold it in place, and that's kind of like a safety measure. You also will need boiling tubes instead of test tubes. Boiling tubes because we're going to be heating one of the reagents up in a water bath, and therefore you'll need a bit of a stronger, thicker, and larger tube so you can actually do that. A pestle and mortar you may need, depending on whether your technicians and your teachers have kindly done this for you at the beginning. I'll explain in a moment. You may not actually use this in class, but in the practical write-up and the method, you definitely need to use that, and I'll explain why in a moment. You, of course, need some food. I mentioned a loaf of bread. It could be a biscuit. It could be some crisps. It just depends on what you want to test and see what types of food molecules are in there. You will also need a white spotting tile. You may need several of these. It depends how many repeats you do. And you will also need your various reagents. So it depends, like I say, what you're trying to look for. If you're looking for protein, if you're looking for lipids, it depends on what you're looking for. So of course, you'll need your chemicals as well to test those various food molecules. So, like I say, you will need to do this step potentially. You might actually have the filtrate ready to use. Your teacher and technicians may have already done this kindly for you. But one of the things you must do is to crush up the food that you're using. So, the sample that you're using, crush it up with a pestle and mortar. And then you're going to use some distilled water and dissolve the molecules of the different food type molecules in that liquid. So the proteins will be dissolved, the glucose, the carbohydrates, the lipids, anything that's present within that type of food will dissolve in the liquid. And you're going to filter that solution, that mixture, using a filter paper and funnel into a conical flask, and anything that drains off all the liquid will contain those nutrients. And of course, depending on the type of food you're using will depend on how much of those are present. And then once you have your filtrate, we can move to the next step. So let's say, for example, we're looking to test for sugar, such as glucose. And in the exam or in textbooks, you might even call this a reducing sugar. Essentially, we're talking about glucose here. We're using a reagent called Benedict's reagent, and this helps us determine how much sugar is present in a sample and whether there's any at all. So you must use heat when you're using Benedict's reagent, just to activate that chemical. You can use a water bath to do this. You can use a you could use a Bunsen burner, but the only reason I would say not to is because if you're doing this practical with other chemicals, specifically ethanol, which tests for lipids, and we'll get onto in a moment, it is incredibly flammable. So I would not recommend using a Bunsen burner or any like direct heat source in that regard. I would use and recommend you use a water bath to gently heat the reagent. Okay, something like 30 degrees, something like that. That will probably be enough of a temperature to do what it needs to do. What you should see, you want to label your boiling tubes. Okay, you want to make sure they're labeled with what is inside them so you know what it is. You also want to use a rack, of course, as well, if you can, to ensure that they're placed in the water bath. You probably want to use a metal one instead of a wooden one for obvious reasons, so that it doesn't get wet, or damaged, I should say. But the results are pretty straightforward. You have three results that you're looking for. So if the food sample has very low amounts of sugar or very low amounts of glucose, it's going to go a green color. Okay. If there's slightly more, there's going to be more of an ambery yellow color. And if there's a lot of sugar, say you're testing biscuits or something like this, you're going to have a very brick red color. Um, One way I've taught this in the past to help students remember is to think of like a traffic light system. Green is very low, orange is kind of medium, and red is like, whoa, there's so much sugar. Okay, red alarm bells. Wow, there's loads of sugar, and that's just one way we've kind of over the years, me and my students, we've kind of remembered it. So hopefully that helps you. Something to mention, the color of Benedict's reagent is a very bright blue. I would say to not worry about the color it was before. So if they ask you what the color change is, you want to say it's gone from blue to green, right? Or you could just say a positive result is a green color. And if there's no sugar, by the way, at all, absolutely none, which is quite unheard of these days with all the sugar that's in food. If there's none, you want to say that the reagent color, there is no color change. You could say it remains blue. Um, that could be another way of saying it as well. So whichever you like to use. Okay, so now we're moving on to another one. If we're testing for starch, now I know this wasn't necessarily in the aim, but this is something you would probably do in your biology lessons as well. So I've fed it in through here. Remember, starch is a form of stored glucose in plants. So you're going to find a lot of starch in very high carbohydrate dense foods, maybe some breads, some pastas, biscuits, vegetables, that kind of thing. So there's going to be a lot of starch stored there. So how do you test for starch? Well, you use iodine, which is like a orangey brown reagent. It's an irritant, so you must wear goggles throughout this entire experiment. And if you spill it, it will stain. You've got to be very careful. Wash your hands thoroughly. But what you're looking for is you're looking for a color change of a blue or black color change, and that tells you there is starch present. And you're using a white spotting tile for this. Okay, you don't need to heat this up like you did with Benedict's reagent. The spotting tile will tell you whether there is starch present, and you want to use a white spotting tile to ensure that you see a very clear color change. If we use, say, a darker colored tile, it would be harder to distinguish that color change. So that is why we use a white spotting tile. Okay, so proteins. How do we test for proteins? Well, we're using a reagent or a chemical called biuret solution. And biuret solution has, again, a very bright blue color. And if there is protein present in a sample, the color will change to a lilac color. And again, we're using a white spotting tile to do this. One way I've taught my students how to remember this is to remember PPP, which stands for positive purple proteins. So a positive result will result in purple or a lilac color, and that is for proteins. That's just one way we've kind of tried to remember in the past. There's so much to remember at GCSE, and I empathize. So if this does help you, feel free to let me know in the comments below. And then, of course, last but not least, if we are testing for lipids or fats, we are going to be using ethanol, which I mentioned earlier. If there is ethanol present, if there is lipids present in a food sample, we are going to see a cloudy residue form, a cloudy layer. And this is very easy to determine. We're going to use a boiling tube for this, or a test tube, even you can use too. We're not heating this up. Remember, ethanol is incredibly flammable. You can also use another reagent called Sudan 3, and it depends on your exam board whether you will need to know this or not. But Sudan 3 is another way of testing for lipids. And essentially, if you place this chemical into a sample, you'll see a red precipitate layer form, and that just tells you that there is some form of lipid. So it's just a bit of extra knowledge for you. So I hope you found that video helpful. Thank you so much for your time. If you did, like I said, join us here on YouTube by subscribing. It'd be great to have you in our community. And thank you if you have already subscribed. I really appreciate your support. You can find more of my revision on Instagram and TikTok at the GCSE Science Teacher. And yeah, I'll catch you in the next one. Have a fantastic day. I've been the GCSE Science Teacher and you have been curious. Take care. Bye.